Thank you very much, Celine, and thanks for the invitation to talk today. So as said, yeah, I work for Chalmers University of Technology, but I'm also a visiting professor at Lund University since May this year. I'm an engineer and a marine biologist from background. And I started off actually after I did my sort of double degree, I went to the US and I worked for the Smithsonian Institution. I was an invasion biologist, we called ourselves. So looking at invasive species in ships' ballast water. And pretty soon I got sick of looking at plankton and started wanting to have some kind of chemical source tracking. So we were interested in ballast water, where it was coming from, whether ships were complying with laws regarding ballast water exchange. And so I started moving into fluorescent spectroscopy as a source tracking for oceanic DOM. And then I did that for a while, and then I did my PhD on that topic in Australia. And then I moved into a postdoc on odor analysis, so untargeted odor analysis. So I was kind of moving around, but you follow the money. And but also I really discovered a great interest in data science. And so I sort of see myself foremost as a data scientist, working primarily with chemical data now in source tracking. And now it's no longer marine environments either. Now I'm source tracking in terrestrial systems. So water treatment, we all know at least a little bit about water treatment. We know that the reason why we do it, especially historically, was to remove microbial risks, so things that can make us ill. Increasingly in recent years, a lot of focus has also become shifted onto chemical risks associated with untreated water. So now we're talking about micropollutants. Some of them are human synthesized micropollutants, and also not only things like that, but also aesthetic problems. When we're talking about drinking water treatment, some of the problems such as taste and odor in those, in the water. The trouble with all of these has been that it's been very difficult to get real-time measurements. So typically to get sort of microbial or chemical data of the right sensitivity, you're talking about pretty specialized offline analyses. And even some of the things online, like flow cytometry, it's difficult to sort of assess the risk. So we have maybe limited information even from the information that we can get. So the question has been, can we have inexpensive proxies of risk? And also, if we have the ability to measure things sensitively through a water treatment plant, then it also gives us the kind of data that we would need to optimize different steps of water treatment. So there's a sequence of steps that occur, and each time we would like to know how well they're functioning and whether we can make them operate more efficiently. So on the left, you're looking at a satellite image of the coast of the Carolinas in the wake of Hurricane Florence in 2018. And the plumes you see are washed down from the hog farms upstream in the storm, and they contain a toxic mixture of heavy metals, fecal contaminants, as well as the organic matter. So often organic matter is an indication of risk, but of course not always. There are organic matters that we're very familiar with that come from, for example, in our cup of tea. So the yellow colour there is related to the compounds in the water that are absorbing and re-emitting, in some cases, light in sort of the brown and yellow colours. So high organic colour indicates a pollution event often, but not always. So as I mentioned, so natural organic matter, we're talking about diverse organic molecules. They may be produced in situ by living organisms. They may be, very often, they're breakdown products. So everything eventually breaks down to the sort of chemical constituents, and those degradation products we're measuring as organic matter in natural systems or in every system. Everywhere that there is water, there is natural organic matter to be measured. It's a useful proxy of risk because both we see sort of the co-occurrence often of microbes, of pollutants, and of high organic matter content, but also in treatment, they tend to be removed together. So they tend to, not completely, but they tend to sort of track each other in treatment. NOM is also something we want to remove because during disinfection, which is like the last stage of drinking water treatment, we might use UV light to try to inactivate the viruses. But the NOM sort of shields those microbes by absorbing the light that we send into the sample. Also, when it's a chemical disinfectant like chlorine, then the NOM reacts with the chlorine. Then also, in addition to making that less effective, there's also the possibility to produce things that we didn't want unintentionally, so byproducts. We're talking about maybe disinfection byproducts, which is typically the category, what we call potentially toxic compounds that are formed through interactions with, say, chlorine. But also maybe smaller fractions of organic matter that are quite bioavailable and contribute to sort of what we call biological instability. So after the treatment is finished, if there's a lot of this lying around, then the microbes can consume it and regrow quickly, say, in the distribution network before the water gets to the customer. So all of this sort of suggests that if we improve NOM removal in water treatment, we're also helping to reduce microbial and chemical risks. In terms of optimization, one of the other important factors is, as you mentioned, Celine, that NOM interacts with all of the different treatment steps. Everywhere it's affecting the efficiency of the process and generally making it harder. Also, we know that there's lots of different fractions, there's lots of different compounds contributing to that signal that we measure as DOM, but they're not equal in their reactivities towards different processes. So this means that if we can efficiently and sensitively diagnose the composition of NOM in water, we should be able to use that information to help predict the reactivity of the organic matter to treatment, and that will help us to optimize treatment. So natural organic matter, if we look at sort of more deeply about it, when we're measuring it, we often pass it through a filter and remove the particulates, and now we've got what we call dissolved organic matter. It's a large pool of which a subset has the property that it's coloured. So as you can see, it sort of appears yellow or brown. And this is measured through absorbance spectroscopy. So we measure the spectrum of the absorbing compounds, and we can take advantage of the fact that if we know what we are measuring, if we control the particular biases, we can measure how much of that absorbing compound is present simply by the signal strength. We also have a smaller fraction, and this fraction, the size is very hard to estimate, but it's maybe on the order of only a few percent, and this is fluorescent organic matter. So some of the compounds or the chemicals that absorb light also are able to re-emit that light at longer wavelengths. So here we're looking at fluorescent spectra. The short wavelength spectra there we call the excitation spectra. It's sort of analogous to the absorbance spectra. But so that this is where it absorbs, but then it re-emits some of that light according to at longer wavelengths, which means at lower energies. And these are independent, so it means we can multiply them together and get a fluorescence fingerprint. So this kind of map here, this is called a excitation emission matrix, or an EEM. And if it's a very simple system, let's say it's this one pure compound, then we have a discrete signal there. But of course, in nature, it's not one compound, it's a whole lot of different compounds that are all present together. And so the signal in nature, of course, is much more complicated. You should ignore the white stripes because these are just where data have been removed because it's affected by artifacts. But just looking at the underneath those white stripes there, what we're looking at is the sum of the fingerprints, if you like, from different chemicals that are co-occurring. And so we're measuring the total. There's a lot of interest in spectroscopy for looking at natural organic matter due to very clear advantages. Those are things such as it's very fast and cheap. You don't have to do a lot of sample handling, perhaps only filtration. And it's not destroying the not destroying the sample at the same time. Also, there's a lot of interest in the fact that we can put this online potentially. So the technology can be put online, so that means we can have continuous monitoring. So continuous feedback in the type of or in the signatures or the patterns that we see in the water over time. And specifically, if we talk about fluorescence, then it has some advantages over absorbance, particularly related to sensitivity. So it's just a way more sensitive technology. If you have a highly fluorescent chemical and it's in a matrix without any interferences, you can measure picomolar concentrations. So it's very potentially sensitive. Of course, we don't have anything like that sensitivity when we have a large background, which is what we see in water treatment. But we have another advantage, and something we call the second order advantage in multivariate data analysis. Essentially, if you take an EAM from one sample and you stack it up with all the other samples that collected, say, from the same system, and if you make the assumption that what you have measured in each of these samples is due to a combination of additive signals, then you can actually go backwards and then decompose the data set into the underlying signals and find out how much of each of those different underlying signals are present in each different sample. And so the most popular way of doing this today is using a model called parallel factor analysis. And then we call something that we obtained, like a fingerprint that we obtained through this method, we call it maybe a parafet component. And the thing to know about a parafet component is this is not without controversy, because there are some environments where the signals don't appear to be additive, particularly under high concentration conditions. So it's a little, so there's been different points of view about this approach, but this is what I've been doing for 20 years. And it's an approach that I believe in, at least under the sort of circumstances that I find myself working in. So these parafet fingerprints then, in theory, they could be pure compounds. This could be the spectral properties of the pure compounds if you went and bought them from from a supplier. Problem is, we don't know what they are. We haven't been able to identify, we've been able to identify probably only one or two of them. So that's difficult. And that makes it very difficult to also assess whether you have a parafet model which is chemically meaningful rather than just some, least square data fitting result. So that has been really a major challenge over the last 20 years, is like trying to decide, are these chemically meaningful and what can we use them for? So let's say we have our parafet model from decomposing a data set. Well, I'm going to put out a hypothesis here, and my hypothesis, I'm actually going to talk about how we get to creating, moving between the model and assessing risk, and also optimizing treatment. And to get this to work, one of the things is you definitely need to have a good parafet model, because this chemical interpretability of the parafet components relies on the model being correct. Otherwise, it's really not necessarily chemicals that you're looking at, or it's maybe a combination of completely different kinds of things that behave in different ways. So, as I sort of mentioned before, it was sort of during my PhD that I made the transition from ecologist to doing chemical data analysis. And at the time, there were very few people actually working with organic matter fluorescence. They were pretty much working always in marine systems. So I was collaborating with Paula Coble because I was doing ballast water, and this is all, of course, marine. And there were a few people around the place, but it was really hard to get the data, and the data analysis was very unsophisticated. So it was people who had looked at a lot of these sort of fluorescence EAMs and sort of characterized very basic features about them, which often were quite good because they had looked at a lot of data. But I wasn't satisfied that we were really getting all the information we could. And so I started working on a toolbox, a software package, to visualize the process of making a paraffect model in order to sort of make good decisions about how you treat the data. And particularly it turned out that, pre-treating the data is a very critical step. If it's not done well, then everything else that follows isn't going to succeed either. And the pre-treating was quite complicated. And so I released this doctoring toolbox in, 2012 or something. And but since then, fortunately, I've worked with, for example, Urban Witch, who was formerly a postdoc in my lab and now is a postdoc at DTU, and we collaborate on keeping this up to date. And he is responsible for the latest developments, which are really quite powerful ways of visualizing the data sets and making good decisions. Also, a problem was that, so paraffect was starting to sort of take off in a kind of an exponential fashion. So all these paraffect models were being published, and people were saying, well, this component looks like that component in that paper. And then you go to the paper and you look at it and go, but does it? Well, first of all, this one's upside down, and to me, it doesn't really look exactly the same. Like, how do we quantify this? So at that time also, I approached a software developer who was working on very nice tools for analyzing GCMS data sets, because I'd discovered this Philip Venig from Lablicate during my postdoc of course, on the ODA data sets. And I said, can you build this database? We want to be able to upload spectra from models that people have created and then statistically compare them so that when we try to identify whether we have a specific component and whether it belongs to a marine system or is it something that comes from the headwaters or something, we want to really accurately be able to compare. And so that was created, and this is called OpenFluor. And today there's about 200 published paraffect models that are in that database. So you can take your model, maybe it's you're still working on it, you can upload it and you can search for statistical matches, and you can go and look at that other paper that seemed to have components that look like yours and figure out, are we in similar systems? Which papers should I be looking at? So this has been a nice tool. And the other thing that we have to a smaller extent is we tried to put some pure compounds, some spectra for pure compounds into the database so that we could potentially match people's models with pure compounds. And as you can see, the red is the pure substance, and then the black are just different matching paraffect models or components that people have uploaded into the database. And you see that for things like tyrosine, there are many very close spectral matches. And this was some time ago, so there's a lot more today. And even like the spectra for salicylic acid, which we know is present in dissolved organic matter, where in a few models, there is a very, very close match for spectra for salicylic acid. But for a lot of the other compounds, we really haven't found matches, and we tend to see sort of evidence of some kind of averaging of signals. So as I mentioned, it has been a bit controversial because some people think that this approach really gives you no information at all. So one of the ways we tried to assess this was to look at whether the spectra that people are producing from paraffect models are actually chemically feasible. So one way to assess that is to measure the distance between the lowest energy excitation peak and the emissions peak, and that's called the Stokes shift. And in real chemicals, that has a limited range in terms of electron volts. And this here is a plot of all the data that were then present in the database. This was 2019 when this one was published. So these points there, like it's from taking this point here as an excitation wavelength, so say 320, and plotting the emission wavelength on this axis, and where that intersects, you get a dot. And then the size of the dot is about how many models have that particular matching information. And what we see some quite distinct clusters, but also a lot of scatter. But we've started to investigate this further, and having done this for a very long time, and having also worked not just on the software end, but also on the experimental design end, how do you get the data from which you should produce a parafact model, I've sort of come to this stage, and my group has come to this stage, of having this hypothesis, which is to a very large extent, the fluorescence components of natural organic matter have predictable spectral properties and nearly ubiquitous distributions. So we will find them whether you're in the deep sea, or whether you're in a river or a lake, and whether you're in the Amazon or whether you're in a boreal wetland, and even Antarctica. So there is a lot of now accumulated data to show that you can have very strongly matching components, and say that a very limited number of components, say often it's 5, will describe 99.9% variability in this data set, but also in that data set. So they can be transferable if they're a good model. If that's the case, if we really have such good information about what that fluorescent background looks like, it opens up the opportunity to also to tell when there's something in the signal, in the whole combined EAM signal, that shouldn't be there, such as a micropollutant which is fluorescence. Now, there aren't a whole lot of really fluorescent micropollutants, but there are some, and we're looking at particularly some sort of pharmaceuticals. So in this this data, so this is collected by my postdoc, Leslie Paradine-Fernandez. You can see an EAM, which comes from river water. And here, that's the same EAM, but this time there's a 5 micrograms per litre of a micropollutant added to it. If you look at the difference, well, there really isn't a lot you can see with your eye without decomposing the data set. But we can assume that we know what should be there and decompose the data set and recognize that certain components should be there, they're natural organic matter, but there are some components really shouldn't be there, and they're micropollutants. And so you can quantify them in a dilution series, say down to, and it looks like that for the ones that we've been working with, it's kind of on the 5 microgram per litre level of sensitivity for drinking water, but also seems to be not that different in the limited wastewater samples that we've been working with. So back here we have our potentially ubiquitous parafact components. And another hypothesis is that to a large extent, the fluorescent components of natural organic matter also have predictable reactivities. So the evidence for this comes from a number of different studies. So a few years ago, I saw that Michael Gonzia and Stephen Timko in the University of Maryland had published a very interesting looking data set. So they were interested in how on the depth variation in the photodegradation of DOM from different sources. They were interested in how pH affects the photodegradation and how it affects the spectral properties. And they collected a very nice data set from mainly Sweden and South America from the Amazon region, also a little bit from North America. And then we ended up supplementing that data set with a sample from Antarctica and a sample from a Tibetan lake and some other places as well. So in the end, I had 15 sort of systems and 35 data sets, all to independently model with parafact, to look at, do we get the same components or different ones? And how do these vary? So they had a very special system that I'll describe first, but I should also say that we also looked at extracts versus natural organic matter, because often we're working with extracts, right, if we're using some other kinds of data analyses. So it's a pertinent question is whether the extract of organic matter has spectral qualities which are similar to the original sample, and also looking at the effect of pH. So here again is the kind of plot where each of the different samples, so the different colours represent these different systems, such as a Swedish lake or this is a deep sea sample. Actually, no, the black one is a deep sea sample. And so what happened is in the lab, these samples were passed through a recirculating reactor system with simulated sunlight and then they were just photodegrading over a period of like 24 hours or a bit longer. And these sort of pictures here are just sort of what the photodegradation sort of looked like for a different parafet component. There's 4 different parafet components. So generally, these were 4-component parafet models. And we model that kind of data typically with a double exponential model where you have a sort of fast rate constant which describes what's happening at the beginning, and you have a slow rate constant that describes what's happening after longer periods of time. And then when we looked at how those rate constants compared across these different systems, so we found that the rate constants were significantly different. So the slow rate constant was significantly different for the different parafet components. But for the slow rate constant, but not the fast one. Here that was more about what was the DOC. So the more concentrated samples degraded faster initially, but then it sort of converged towards whatever the parafet component was going to do. So we ended up having very similar parafet components from each of these systems, but also degrading at very similar rates. Also looked at in another study in their reactivity to biodegradation. So this was a study by a PhD student who finished a couple of years ago, Nishina Muna. We have a very nice cooperation with a water treatment plant in Gothenburg. And they have a biofilter at the very first step of the water treatment. So it comes from the River Elbe and goes straight through this sort of biofilter. And the biofilter is an expanded clay media. So it's designed so that it has lots of little nooks and crannies for microbes to hide in so that it can be a good biodegradation effect. Then they very kindly changed the rates that the water was flowing through each of the filters so that the 4 parallel filters all had different filter rates. And so that's what you're looking at, flow rates. What you're looking at here, where you have 4 different bars for different sort of contact times. But what we see also is that only a couple of those components are at all reactive to biodegradation. One of them is one that has long been known. But then here's another one as well that's very much seems to be a biological a tracer of bioavailable organic matter. Then the other components, they weren't actually being removed at all. They were actually being sort of released from the filter. We've also been looking at their reactivity to chemical water treatment. And for a long time, people have discussed a parameter in water treatment called SUVA, and that is the specific UV absorbance. So you measure UV at a particular wavelength. It's usually 254 nanometers, mostly by convention. And then it's normalized to the DOC content. And from this, you can find out if it's a relatively highly absorbing, so highly sort of an aromatic sample, it's dominated by aromatic compounds, or whether it's dominated by less aromatic compounds. So this SUVA parameter has been quite interesting because also it's correlated to things like some disinfection byproducts. How many are produced can be predicted from the SUVA value. So SUVA has been long known to be a sort of a proxy, not a perfect one, but a proxy of some aspects of reactivity of NOM. But it's a bit of a pain to measure. And it's the absorbance, it's 2 different instruments to start with. And so as instruments each have little artifacts that can translate to those artifacts that are multiplying when you compare between different labs. Also DOC analysis, for anyone who's been involved with it, knows that it's quite sensitive to analytical interferences, to contamination. So in some ways, it's kind of an underutilized parameter, perhaps because it's just not the kind of thing that a water treatment plant can just easily get data for at all. It's offline and it's relatively expensive. It's not part of the package that they would normally pay for to get measurements of water. So what we did find though, and so in this study, I worked with Marc Philibert at Suez in Paris, and of course Suez is a major water distributor or treatment company, and of course they have a global reach, and they therefore were able to collect samples from many different water treatment plants, so around the world, and also from different stages of water treatment plants, and bring this back. And then I formed a Parafac model for this data set, and we had a look at how these varied across the system, and also in relation to other kinds of data that they also collected. And what we found was that we could get a very good proxy of SUVA by measuring just the ratio of two Parafac components. And that ratio is called our Parix, or the Parafac index. Another thing that was interesting and also supports what I say about SUVA being a kind of a tricky parameter is that we got better correlations when the SUVA was measured by LC-OCD compared to when it was measured by the standard method with two different instruments. So this would be very nice because fluorescence is much easier to measure, it's more precise, and then it's potentially a way that we can get a proxy of SUVA and also of reactivity. But then to prove the reactivity part of it, they also took samples from 5 different rivers in France. They had different DOC and different SUVA values. And they subjected each of these water to a suite of like 10 standard tests. So they're all subject to the same test, and looked at how well DOC was removed. And then DOC removal was, for some of these tests, for example, for coagulation and ozonation, was very well predicted by this Parafac index. So what's significant about this is to remember that for fluorescence, we're talking about a very, very small fraction of the organic matter, but we're measuring the removal of all of the organic matter that we can quantify with DOC. So even though it's a small component, it contains a lot of information that's useful for telling us about the other parts of the organic matter as well. And also these are other Parafac components and other ratios that also were more predictive for, say, powdered activated carbon or a combination of PAC and ozone. So I'm going to summarize where I've come to now. So this comes from the idea that NOM, first, is a useful proxy of risk in terms of microbial and chemical risk in water treatment, so that it's worth using NOM in order to help optimize and assess risk. But to do this, we have to be able to detect when the quality of water and its reactivity has changed. We have to be able to do this very sensitively. So a hypothesis is that most of the measured fluorescence in most of the systems that I've worked with, now I'm talking about mainly freshwater systems, drinking water. I haven't done a lot of work in wastewater, and I know that these are more complex matrices, but that's a sort of area that I'm interested in moving into. So we have these widely distributed components, but we're also finding some preliminary indications that they're not too bad when you're looking at wastewater as well, which means that sort of a reliable Parafac model can be transferable to another system. And we can maybe use that information if we have these reliable Parafac models. We know a pretty good idea about some of these components that should be present, to then also expose things that shouldn't be present, such as fluorescence micropollutants. The fluorescence components, they vary in their reactivities towards different treatment processes, both chemical and biological, and we can use the ratios between some of those fluorescence components to tell us something, to predict how well a treatment is going to work for a particular composition of water. I've come to the end of my talk. So I just finished by acknowledging that there are many people involved in, or have been, many people involved in all of the research that I've showed you. These are some of the people who've contributed the most to my presentation, so I thank them. I thank you all for listening, and to Eawag and Celine for the invitation to speak to you today. Thanks.